On to Chapter 17, Part 2, Breathing Mechanics. We're going to do a little bit of VQ ratio today, along with some more phys in the physiology. The lungs are actually a surprising array of different types of tissue. You certainly have epithelial tissue, but you also have extensive connective tissue, smooth muscle, vascular tissue, lymphatic tissue, and nerves. It's a very complex system given that most of the time we think of them as just relatively glorified air sacs. We regulate how air moves in and out of the lungs, not only in terms of in and out, but how much. We can modulate airway resistance. Oftentimes we think of this in terms of pathological reasons, but there are also physiological reasons for modulating resistance, just like in the vascular system. We have two concepts that we're going to be talking about a little bit. One is called compliance, and the other is called elastance. And compliance is the ability to stretch. Now, again, like all things in physiology, there is a homeostatic window for compliance. You can have too little compliance, like in the case of pulmonary fibrosis, or you can have too much compliance in the case of someone who has a flail chest or a premature infant. We also have elastance, which is the ability not only to stretch, which is compliance, but elastance is the ability to return back to the original shape. It's the ability to return to its original size after being stretched. Make sure you get those terms down. Compliance is the stretchability, the expansibility, and elastance is the ability to return to its original shape and size. For instance, if you have a piece of chewing gum that you've chewed on for a while, it's plenty compliant if you stretch it in your fingers, but once you stretch it, it's not going to return back to its original shape. It has low elastance. Then we have a concept that many of you are probably not as familiar with, which is called the VQ ratio, ventilation relative to perfusion. The Q is perfusion or blood circulation. And we regulate blood circulation in the lungs as much or more than we regulate airway resistance. What is VQ ratio? Under normal circumstances, we define VQ ratio as a 1-to-1. It's the amount of ventilation and perfusion in the lungs to meet metabolic need for gas exchange delivery to tissues. If we have a low VQ, that means that we do not have enough ventilation relative to the blood flow, and so the blood is not going to be oxygenated or decarboxylated properly. If we have a high VQ ratio, we run the risk of alkalosis because we have a lot of ventilation relative to perfusion. So the blood is certainly being saturated in oxygen, but you may actually be depleting carbon dioxide. And then there are scenarios where you have a V of 0 or a Q of 0. Those are a little bit beyond this course, but I want you to understand what it means in terms of the VQ ratio. So when ventilation exceeds perfusion, you're breathing more than you can possibly oxygenate that blood. Examples of this involve COPD and so forth, but again, some issues may also be involving with alterations of pH. Poor ventilation, we have lack of oxygen supply. That can be bronchitis or asthma, pulmonary edema, where we're just not getting that oxygen to the blood and we're just not getting that carbon dioxide out of the blood. And then we have things like shunt, like for instance, if we have no air entering the alveolus, like in the case of an occlusion or an obstruction, okay, pneumonia, a condition called atelectasis, or we have no blood supply to the lung, like in the case of a pulmonary embolism, that sort of a thing. So you have plenty of ventilation, but you're not getting blood there. So these are all examples of applications of the VQ ratio, but I want you to understand what the concept is first. Now, if we go down to the alveolus, the level of the alveolus, if you remember in the couple of slides previously, I said that the lungs are a surprisingly complex tissue system. Well, at the alveolus, we don't have quite as many tissues. We have basically 4 major cell types because the alveolar structure is fundamentally about exchange. So we're not going to want a whole lot of cells and layers and that sort of thing because we need a short diffusional distance. I want you to look down here in the lower right corner here, right here, or perhaps right here. You can see that there are actually two cell layers right here. That's the typical distance between the alveolar space and the blood plasma. It's got to be that small because we're talking about diffusion here. And so diffusion is proportional to distance. And here's another example of it right here. Diffusion is proportional distance. So we have to have a very short distance if we want a high rate of diffusion. So what cell types do we have in here? The most numerous cell type that we have is called the type 1 alveolar cell, a simple squamous epithelial cell, very, very thin exchange epithelia. That's the most numerous, and that's the one you typically see if you had a section of lung under the microscope. The next cell I want to talk about is the type 2, Roman numeral 2, alveolar cell. These are not exchange epithelia, these are secretory epithelia. And what they are doing is secreting a mixture of lipids and proteins and water to create a mixture known as surfactant, lung surfactant. The diameter of the alveolus and alveoli are so small that the surface tension actually increases. There's something known as the law of Laplace we'll talk about later on. Because they're so small, there's high surface tension, which means that water will tend to ball up into droplets. And water has enough cohesive force within those droplets that it could quickly fill up alveoli very, very, very quickly. You can do an experiment on this. Go to your nearest faucet, rinse your hand with some water, as you should be doing several times a day, by the way, but don't dry your hand off right away. Just hold your hand up and let the water drip off of your fingers. You can easily see those water droplets, and those water droplets are huge, are massive compared to the size of alveoli. So if water can hold itself together in a droplet that large, it can easily fill up these alveolar spaces. And so surfactant becomes essential into breaking up. They call it a wetting agent, if you will. It causes the water droplets to no longer be able to form. And so it creates a very, very thin layer of water on the interior surface of the alveolus. The third cell type that we have are modified white blood cells. Here's one down here at the bottom. Here's one up here. These are macrophages. These are modified white blood cells that have exited the vascular system in the lungs by a process known as diapedesis. Diapedesis is the migration of a cell to other spaces, and these macrophages have moved out in and around the alveoli outside of the capillaries. Just like the microglia in the neurologic system, these guys act as Roombas inside and around the alveolar spaces, phagocytosing potential irritants, debris, dead cells, potential pathogens. Macrophages are implicated in the types of cancer that we see associated with asbestos. What happens is these guys phagocytose the asbestos needles, and then that ruptures the macrophages and triggers inflammation. And so you have this chronic inflammation in the lungs that then is pro-mitotic and pro-mutagenic, which leads to certain types of lung cancer associated with asbestosis. The fourth cell type is another epithelial cell. These are the endothelial cells that we talked about in the vascular system. These are the capillaries. So again, very, very thin, very short distances between the plasma space, the plasma compartment, and the alveolar air compartment. So this slide is meant to reinforce something we talked about in Unit 1, which is the idea of diffusional distance proportional to rate of diffusion. So we have to have very, very short distances in order for diffusion to be rapid enough to meet metabolic need. And this distance between the blood plasma and the alveolar airspace can be anywhere from 1/10 of a micron to 1.5 microns. And just to put it in perspective, 1/10 of a micron would be roughly 15 times faster diffusion, net diffusion, than the 1.5. 1.5 really isn't all that much, but again, it has enough that you could have a 15-fold difference between these two distances. So imagine what could happen if we all of a sudden had pneumonia or inflammation leading to additional fluid. All of a sudden, instead of being 0.1 to 1.5, now we're 8, 10, 12 microns or more. That could effectively shut down diffusion entirely, or so low to the point that you can no longer properly oxygenate and decarboxylate the blood. Okay, now it's time for the gas laws, and I will already know that there's a lot of math going on here and a lot of things going on. First of all, I'm going to try to couch these concepts in ways that won't require that you need a calculator. I think it's silly that we are so reliant on calculators that we can't do regular back of the napkin math. But more important than being able to calculate a number is to understand why that number would be important, or what that mathematical ratio actually means. All the numbers in the world are pointless if we don't have meaning to them. So I want to work through these sort of three big things: the ideal gas law, the Boyle's law, and Dalton's law. Ideal gas law I have found in my life, since I like to scuba dive and I like to fly and I like to climb mountains and so forth, I've actually found it to be one of the most useful formulas for doing things that are just cool. You know, you have to sort of understand gas laws in order to be safe as a scuba diver. You certainly have to understand gas laws if you want to be involved in the respiratory care and the appropriate health care of somebody. Certainly flying and climbing and altitude issues are all part of that as well. So PV equals nRT. Pressure times volume is equal to the number of molecules, or the amount of gas, times a correcting constant times temperature. So what does that all mean? Well, the first thing I would tell you is that if we keep everything constant, if we keep the number of molecules of gas constant, if we keep the temperature constant, we can do a little bit of algebra, and what you find is that volume is inverse to pressure. Volume is inverse to pressure. Another thing that I would like to get out of the way right now, and I'm going to get rid of that box there real quick. I want you to look down here, and this is actually Boyle's law. We'll get to that in a second, but I want you to get this concept right now. Because gases all obey the ideal gas equation, all gases obey the ideal gas equation, one of the things you can figure out is that pressure is synonymous to concentration. Pressure is synonymous to concentration. You can have 1, 2, 3, 4, 5 molecules of gas here in a given volume, like for instance, 5 molecules per liter. Okay? What is concentration? Is number of molecules per unit volume. So let's say this is 5 molecules per liter. Well, let's keep those same molecules, same number of molecules, 1, 2, 3, 4, 5, only this time let's make it half a liter in volume. Well, now we have 1, 2, 3, 4, 5 in half a liter of volume, which is the equivalent of 10 molecules per liter. So pressure is synonymous with concentration. If you can get that, more than anything, the gas laws will begin to make sense. Okay, so get those two concepts, that volume is inverse to pressure. That's because pressure is synonymous to concentration. Everything else will fall into place. Hopefully, okay? But if you can get those two things, you've gone a long way to really understanding the point of gas laws. Okay? So let's get into, I'm going to do probably what is the trickier of the two gas laws. It's known as Dalton's Law. And if you were to crunch the numbers and you were to compare PV equals nRT and do all that fun stuff, what you would find is that you can derive Dalton's law algebraically. But here's what Dalton's law says. It says that if you have a pressure of a mixture of gases, that total pressure is the sum of the individual pressures of each gas, also known as the partial pressure. So let's do a simple gas mixture real quick and a simple pressure. Let's say we have 100 millimeters of mercury of a gas mixture, two gases, equal proportions of both gases. So we have two gases, equal proportions. That means 50%, 50%. So 50% of 100 millimeters of mercury would be a partial pressure of 50 millimeters of mercury. And the other gas would be also 50 millimeters of mercury. This is the beauty of the ideal gas law, is that gases behave the same way. Even if they have different reactivities, different densities, and so forth, they still behave under the ideal gas law. So we have the sum of the partial pressures. So let's take it to the next level, and let's just do atmospheric air. Understand that for testing purposes, I want you to understand the concept of partial pressures. Not so much being able to do some complicated math. I'm not going to ask you to do anything other than back of the napkin math with this stuff. But let's just do regular atmospheric air, shall we? So atmospheric air has an average pressure of 760 millimeters of mercury. That's the average atmospheric pressure. It goes up, it goes down, depending on the weather and so forth, but the average is 760. Atmospheric air, no matter where you are on the planet. I know you might think, well, what about at the top of a mountain and so forth, or that sort of a thing when there's less oxygen? That's true, but it's because there's less pressure. It's still 21% at the top of Mount Everest. It is still 21% at the bottom of the Dead Sea. So if we have 760, which is our average, and we multiply it by 0.21, 21%, that gives us a partial pressure of oxygen, 21% oxygen. 0.21 times 760 is 160. Okay? So it's over here. So we have 160 millimeters of mercury because it's 21% oxygen. Carbon dioxide's much lower percentage. It's only 0.03%. So if we multiply 760 by 0.0003, we have 0.25 millimeters of mercury of carbon dioxide. So using Dalton's law, you can calculate the total pressure of a gas from the sum of all the partial pressures, but you can also figure out the partial pressure of gas based on the percentage times the total pressure. Now, where does this all go? Why are we learning all this? Let me give you a magic number, and I don't have it written down in this slide, but you should just go ahead and write it down because this has huge therapeutic implications. What we want to do at any circumstance, whether you're at the top of Mount Everest or the bottom of the Dead Sea, somebody you're trying to keep alive, the magic number therapeutically and physiologically for oxygen inside the alveoli, we want a pressure, a partial pressure of oxygen to be 100 millimeters of mercury. So somebody who's doing a deep scuba diving dive down to the bottom of a really deep, pit, underwater pit or something. What they do is they vary the percentage of gases so that the oxygen in the lungs, regardless of pressure, remains at or around 100 millimeters of mercury. The deeper you go, the lower the percentage of oxygen you want, because you want to lower that percentage because pressure is rising. And pressure is synonymous with concentration. Likewise, if you want to go to the top of Mount Everest, it's probably a good idea that you bring some oxygen along with you because the partial pressure of oxygen is very, very low. And so you can no longer sustain 100 millimeters of mercury just breathing atmospheric air. So what do? You breathe additional oxygen. The higher you go, the more and more percentage oxygen you need in order to sustain that 100 millimeters of mercury. That's why folks that go to super duper high altitudes, like, flying the SR-71 or going into space and so forth, wear those spacesuits. Part of the reason why they wear those spacesuits is to keep atmospheric pressure to a point where you can achieve 100 millimeters of mercury of oxygen in your lungs. 100 millimeters of mercury of oxygen, that's the magic number. Like pH is 7.4, temperature of 37 degrees Celsius. Boyle's law is a lot simpler, and I've already sort of explained it to you. Mathematically, if you take out temperature, if you keep temperature constant, you keep number of molecules of gas constant, what you will find is that pressure is inverse to volume. That is to say, if we multiply pressure in volume in container 1 times volume in container 1, that product will be the same as pressure in volume 2 times the volume in container 2. So let's see, 1, 2, 3, 4, 5, that would be 5 molecules per liter. They're defining that as 100 millimeters of mercury. We're not worried about that. In any case, when we cut the volume in half, assuming temperature is constant, number of gas molecules is constant, we now have double the pressure. Pressure and concentration are synonymous when it comes to gases. So we've talked about the laws, the gas laws, that is to say the ideal gas law, Dalton's law, and Boyle's law. Now let's start applying some of those laws into the anatomy and the movement of air, the process we know of as ventilation. And that requires air flow. So a little bit of convention for you. The first thing is that airflow, when we talk about a single cycle of respiration, it begins with one inspiration followed by one expiration. Typically, we use a little bit of muscle. That's the diaphragm. The diaphragm muscle contracts during inspiration, usually not using the intercostal or accessory muscles at that point, followed by a relatively passive expiration. And so we have to have air flow. And flow, just like in the bloodstream, is a process of pressure gradients inversely proportional to resistance. So we have a change in pressure. We lower the pressure in the lungs when we inhale. That causes air to equilibrate. And we increase the pressure when we exhale, causing, again, an equilibration. But it's inversely proportional to resistance. Where resistance really plays a role is in pathophysiology. Under normal circumstances, we have a nice open airway and there's relatively little resistance going in. But if we have a constriction of any kind, like for instance asthma, where we have a chronic inflammatory situation with occasional acute attacks, or in the case of pediatrics where they just have smaller tubes to begin with, remember that it's radius to the fourth power when it comes to resistance. So a child that has a radius that's half of an adult's would mean 1/16 of the flow. So that's a big reason why upper respiratory infections tend to be much more serious in pediatric cases. And so forth and so forth. So we do have those same issues of pressure versus resistance in our airways. We do have passive expiration. I've already mentioned that, and that has to do with elastance. Active expiration is where you're actually contracting accessory muscles and intercostal muscles in order to force the air out. Usually that active expiration is due to either increased metabolic need or you're trying to expectorate something that's occluding the airway of somehow. A cough is a common example of active expiration. When it comes to airflow in the lungs and the pulmonary system, we can measure two things. We can measure rate and we can measure volume. Spirometry can do both. A spirometer is basically a machine that is designed to trace not only the overall volume, but the rate of change of volume. That is to say, if you have a slower rate of change, you would see a shallower curve. Faster rate of change, steeper curves, and so forth. Right now, I'm going to talk primarily about volume. I don't really get into forced expiratory volume and rate all that much in this class, mostly for time purposes. I'm already requiring a lot of your time as it is, and it feels like a lot of material. So I'm just going to focus on volume in this lecture. So let's break down some of the key volumes that are involved in understanding lung function, and by extension, if we can move air, we imply that we can exchange gas. This is called a spirometer tracing. So we have to have a reference point. We have to have a reference point. And we set that reference point right here at the end of a normal exhalation, an involuntary exhalation. So the typical breath, we inhale and we exhale. Inhale, and then we passively exhale. This up and down in the normal resting pattern, roughly about half a liter, is known as tidal volume. The tide comes in, the tide goes out. The tide comes in, the tide goes out. About half a liter under normal resting circumstances. Again, we define it at the end of exhalation, normal exhalation. Now, if we inhale in and then we decide to inhale as far as we can above and beyond tidal volume, all the way in, what we have there is known as inspiratory reserve volume. So we inhale about 500 milliliters, and then anything above and beyond that 500 is considered inspiratory reserve, to the tune of almost 3 liters or even more in people who exercise their lungs or happen to have large lung capacities. People like those professional bikers are basically lungs with a pair of legs. So anything we can draw in beyond a normal inhalation is inspiratory reserve volume. And that's huge. It's 6 times the normal tidal volume. Why is it so big? Because there are times when we have intense metabolic need under fight and flight. It's strenuous exercise, metabolic need. We need lots of oxygen. So we have a lot of inspiratory reserve volume. Now, if we add IRV and tidal volume together, we have 3,500 milliliters. That gives us inspiratory capacity. That is how much we can inhale from the bottom of a normal breath. So I breathe out and then all the way in, 3,500 milliliters or more. That's known as inspiratory capacity. It's the adding of the IRV plus the tidal volume. So now let's switch down to the lower side of this, and remember that our benchmark is again the end of a normal exhalation. But if we breathe out beyond that, the way out, okay, about 1.5 liters, that is expiratory reserve volume. 1.1. I'm not going to worry about the numbers for you guys. I want you to be able to recognize these volumes from a chart. So we have expiratory reserve volume, that which we can exhale beyond a normal involuntary exhalation. Okay? Normally, exhalation is a recoil. What we rely primarily on elastance to drive a normal tidal volume exhalation. To do ERV, you have to use accessory muscles and intercostal muscles and that sort of thing. Same thing's true with IRV. You use your diaphragm primarily for tidal volume, and then you start using accessory muscles and intercostals in order to achieve that reserve volume. If we add all these together, if we add ERV, if we add tidal volume, if we add inspiratory reserve volume, IRV, from the bottom of your furthest out breath to the top of your furthest inhale, that is known as vital capacity. So it's 3,000 plus 5,000 plus 1,100, which gives us a total vital capacity in this person of 4,600 milliliters, almost 5 liters, getting close. Again, if you stretch, and you work those lungs out, and you're in good shape, and you are so forth, you can increase your vital capacity and thus increase your ability to oxygenate and decarboxylate your blood. Just one more volume here. I know I've been going for a little while on this slide, and that is to say that the lungs are also made up of tissue and pipes and blood and connective tissue and all sorts of things. That is known as residual volume. This is not true functional volume. This is just the space occupied by material in the lungs. Okay, so residual volume is just, it no longer involves air movement. It's just the space occupied by tissues and structures. This is just another picture of that same thing, showing you the different volumes: tidal volume, inspiratory reserve volume, inspiratory capacity, expiratory reserve volume, vital capacity. One thing and residual volume down here. One thing we're not going to test you on or worry about is functional residual capacity. I really don't know what the significance of that is. It probably has to do with fibrosis or other types of issues that alter the amount of residual volume and thus affect the ability to exhale. Well, we've hit the 30-minute mark at this point, and again, I have about 12 or 13 more slides that I'd like to cover, so I'm going to stop right here since we've hit 30 minutes, and I will talk to you again in Chapter 17, Part 3. Thanks for your time, and I'll talk to you soon.